Hello, and welcome to this short video where we will introduce subsequent entry biologic drugs and how they relate to treating anemia of chronic kidney disease. First, we will discuss anemia of chronic kidney disease and its treatment. Then, we will introduce biologic drugs. And finally, we will answer the question of what subsequent entry biologic drugs are. Let's start with anemia of chronic kidney disease and its treatment. Anemia can have many causes, and one common cause is having low kidney function. When kidneys are healthy, they produce a hormone called erythropoietin, or EPO. EPO prompts the bone marrow to make red blood cells, which then carry oxygen throughout the body. When the kidneys are diseased or damaged, such as when someone has chronic kidney disease, they no longer make enough EPO. As a result, the bone marrow makes fewer red blood cells, causing anemia. Treatments for anemia of chronic kidney disease include iron supplementation and injections of genetically engineered erythropoiesis-stimulating agents, or ESAs. A large portion of people with anemia of chronic kidney disease require these injections. Now, we will move on to discuss biologic drugs and their relevance to treating anemia of chronic kidney disease. Biologic drugs are a special category of drugs that can be made up of large molecules such as sugars, proteins, nucleic acids, or combinations of these substances. They are isolated and produced from living organisms and are used to treat many medical conditions including diabetes, anemia, and autoimmune diseases. EPO, used to treat anemia of chronic kidney disease, or insulin, used to manage diabetes, are examples of biologic drugs. As you may have guessed, biologics are much more difficult to produce compared to traditional drugs with a well-defined chemical structure such as aspirin. This slide shows a side-by-side comparison of a small drug, aspirin on the left, versus a small biologic protein, such as erythropoietin in the center, and a large biologic antibody on the right. The structure of a biologic drug is several times more complicated than a drug such as aspirin. It also takes many steps to create a biologic, and because they are made by living cell cultures, the manufacturing process is very delicate. A small change in a single step can change the structure, and therefore the properties, of a biologic drug. As you can imagine from their complexity to produce, biologics come at a cost. In British Columbia, it is estimated that an average of $6,000 is spent each year on each patient who requires an erythropoiesis-stimulating agent. Public and private payers predict that expenditure on biologics will increase over time as they are used to treat more diseases and as more products come onto the market. Authorities are therefore looking for ways to balance costs while improving access to these treatments. Subsequent entry biologics, or SEBs, are one solution, as they are biologic drugs which cost less than the original or innovator biologic product. One study has estimated that an EPO SEB could save the Canadian healthcare system $220 million in the first 5 years of their use. This table describes some general ways in which an innovator biologic and a subsequent entry biologic compare to one another. An innovator biologic will be an original product, while an SEB is a biologic which is similar to an innovator biologic. It also has other names, such as biosimilar or follow-on biologic. The innovator biologic is the first to receive approval. while an SEB will enter the market subsequent to an approved innovator biologic when the innovator's patent expires. Innovator biologics will undergo original and extensive research and development, including full efficacy and safety testing, while subsequent entry biologics will undergo an abbreviated process for approval. You can see here that an SEB is created to be as close as possible to an innovator biologic, but because of the delicate process by which biologics are made, they are not exact copies. People often question if SEBs are considered generics. While both classes of drugs have emerged due to patent expiry of brand name drugs, there are very clear differences such that SEBs should not be considered generic biologics. Some of these differences are as follows. Generic drugs will be a small molecule drug, while SEBs are a large complex molecule. Generic drugs will be chemically synthesized, while SEBs are manufactured in a living system. A generic drug will have an active ingredient that is chemically identical to the reference product, while an SEB will have an active ingredient that is highly similar to the reference product. And finally, generic drugs are approved on the basis of analytical similarity and bioequivalence, a pharmacokinetic study, while subsequent entry biologics are approved on the basis of extensive in vitro comparability testing and reduced non-clinical and clinical comparability testing, such as toxicity, pharmacokinetics, safety, and efficacy, compared to their innovator biologic. In summary, SEBs are more difficult to produce than generics, and therefore they require more extensive testing before they are approved for use. So we can see that SEBs must show that they are similar to their innovator biologic before obtaining approval by Health Canada. However, as Health Canada considers SEBs a standalone product, no other assessments of similarity need to be done between the innovator and the SEB after the drug is approved. This could potentially result in the innovator and SEB drifting apart or becoming closer in terms of similarity, perhaps as the result of a manufacturing change. When it happens, it is known as drift. So what is the context for using SEBs in anemia of chronic kidney disease? This has become relevant in Canada because the patents on the drugs that are used to treat anemia of chronic kidney disease have already expired. Other patents on other biologic drugs have also expired in Canada, meaning that SEBs may soon enter the market in this country. Patent expiry on ESAs has been very recent in Canada, but in Europe, there is almost 10 years of experience with EPO SEBs. So far, post-marketing research and surveillance has found ESA SEBs to have similar efficacy and safety to the innovator drug. SEBs have also been found to reduce cost, which can be a major barrier to accessing medical treatment. However, there are also potential risks with their use, and this all centers around how complicated it is to produce biologics.